The last lecture covered the prokaryotic groups, the bacteria and the archaea. Now we're going to move on to the eukaryotes. Eukaryotic cells come in an amazing variety of shapes and sizes. They're incredibly ecologically diverse as well. Most of the time that we spend in the rest of this class will be spent on the large, the multicellular groups within the eukarya. But today's lecture is going to be spent on some of the groups you hear less about. These are known collectively as the protists, and they're small, usually unicellular eukaryotes. Before talking about the protists specifically, what I'd like to do is talk a little bit about how eukaryotic cells are different from prokaryotic cells. Most notably, the eukaryotic cells contain this organelle, this membrane-bound organelle in the center of the cell called the nucleus. In fact, that's what the word eukaryote means. It means true nucleus. There are also a large number of other membrane-bound organelles within a eukaryotic cell. There are histone proteins associated with the DNA, which occurs in a very different form than it does in bacteria. In the eukaryotes, the chromosomes are linear, which allows them to be copied much more efficiently than the circular chromosome of the bacteria or the archaea. In addition, there is an extensive cytoskeleton that exists within eukaryotic cells. It's made up of a number of elements that you can see here. One of them is filaments of actin. Actin, as you may remember, is one of the structures or one of the filaments that makes your muscles contract. There are also microtubules made of tubulin, and these filaments made of strong protein fibers. All of these are woven together into a structure that not only supports the cell, but can be remodeled to change its shape very dynamically. This scaffolding extends all throughout the interior of the cell and is used to move membranes around as well as moving the outside of the cell around, so that the entire cell can take on a myriad of different shapes, some of which are very important for protist function. For example, the fact that the membranes of the cell can be remodeled dynamically allows things like this amoeba to move around and engulf its food, bringing it into a vacuole which is then pinched off in the interior of the cell for digestion. Bacteria and archaea can't do this in any way, partly because they lack the cytoskeleton necessary to dynamically change the shape of the cell, but also because they're encased within a rigid cell wall. Most of the eukaryotes do not have a rigid cell wall, and so they're capable of moving in ways and behaving in ways that cells that are bounded with the cell wall simply can't. However, the biggest difference between eukaryotic cells and prokaryotic cells is that eukaryotic cells are simply much, much bigger. In terms of volume, they're about 1,000 times bigger than your average prokaryotic cell. And as I've tried to hammer home by talking about allometry in this class, that change in size makes a very big difference. One of the things that it particularly makes a difference for is transport. Diffusion in and out of a cell that's as large as a eukaryotic cell is going to be much less effective at getting this cell's needs met than it would be for this smaller prokaryotic cell. In fact, the movement of substances within a eukaryotic cell moves very slowly, just because the cell is such a big structure relative to the rate of diffusion. One of the ways that eukaryotic cells have evolved to deal with this problem is that they have this complex set of internal membranes, which are used to partition parts of the cell from one another and localize certain reactions and processes, so that instead of having them spread out across the entire cell and having, say, an important enzyme at very low concentration, you can maintain an enzyme at high concentration within one of these compartments, and that's where the reactions it participates in take place. In addition to compartmentalizing function, these membranes present a lot of surface area for reactions and processes that require some sort of surface. So the internal membranes are a big step forward in terms of allowing eukaryotic cells to do novel things. The protists, which we're going to be talking about today, are not a really good group, as taxonomists call it. They're a grab bag. Basically, they are all the eukaryotic organisms that are not plants or animals or fungi. Makes them a paraphyletic taxon. And it also makes them very difficult to talk about, because really you can only talk about them in terms of what they are not. Other than not being plants and animals and fungi, and other than mostly being found in moist environments, there is not a whole lot that all protists have in common. They're all eukaryotes, but some of them are multicellular. Some of them are tremendously large. Some of the multicellular protists can be 60 meters long. Most of them are so small, however, that you can't see them with the naked eye. They make their living in a variety of different ways. They're parasites, predators, but also a tremendous number of important producers. They can move around. Some of them have hard shells on the outside. Some of them have hard shells within their membranes. And some of them, like the amoeba, can have whatever shape they want. It's hard to find a lot in common that you can say about all protists. However, protists do have some very important effects on us. So, for example, throughout human history, they have been the cause of some severe problems that human beings have had. The Irish potato famine was caused by an oomycete, a fungus-like protist that killed off massive numbers of potatoes in Ireland at a time when the country basically depended on them for food. Worse is malaria, another protist that is caused by the species in the genus Plasmodium. It lives within blood cells and kills over 1 million people worldwide every year. Somewhat more benign are protists that cause red tides. These are basically population booms of certain photosynthetic protists. The problem with these photosynthetic protists is they secrete toxins into the water. And when these are concentrated enough, you have a condition that is very dangerous, because shellfish, things like oysters and mussels, filter food out of that water. They will concentrate the toxins in their own tissues, and if you eat these contaminated shellfish, they can actually kill you. But really, the biggest impact that protists have on us is in their role as part of functioning ecological systems. They are extremely abundant. A teaspoon of pond water contains 1,000 flagellate protists. Those are protists that move themselves around by waving flagella. Dinoflagellates can be found at concentrations of up to 60 million in a single liter of water. In the oceans, diatoms are protists that are among the leading primary producers. And because the world's oceans are so large, that production, even though it's very small on a per meter squared basis, the total production of all of those producers in the ocean contributes more to net primary production on this Earth than does the production in any other biome. Protists, along with other members of the plankton, are the basis for every food chain in the oceans. So a tremendous amount of Earth's biomass ultimately depends on the ability of protists to generate food. Without them, most of the oceanic food webs would completely collapse. There are a number of things that are very curious about protists. As I mentioned, they have a lot of intracellular membranes, including some compartments that are considered to be intracellular organelles. But a few of these intracellular organelles have a very strange origin, which has now been reasonably well proven. It started when it was first proposed by Lynn Margulis. Um, it was thought to be such an absurd idea that no one took it seriously. But by now, so much evidence is accumulated supporting it that it's pretty well taken as fact. Mitochondria are organelles that are found in essentially all eukaryotic cells. Even the ones that don't have them today apparently once had them in their evolutionary history. They have double membranes, and these are called the powerhouses of the cell because they are they're the location where oxidative respiration occurs in cells, providing us with all the ATP that our cells require to live. Chloroplasts are in some ways similar in that they have a double membrane. They're not found in all eukaryotic cells, but they're found in a wide variety of them. The origin of the eukaryotic, the origin of the mitochondria in the eukaryotic cell is debated somewhat. But the one thing that is not debated anymore is that the mitochondrion used to be a free-living aerobic proteobacterium. That is, it was a bacterium that could use oxygen in its own metabolism. Somehow this bacterium, this free-living organism, found itself living inside a larger cell of another species. And maybe it was a parasite living within that cell, maybe it was engulfed by the larger cell but not digested. No one really knows. At any rate, the end state was that you have these larger cells with organelles within them that are generating energy. These larger cells feed high-energy carbon compounds to their guests, and the guests in turn use oxygen to generate very large amounts of ATP, which is then shared with the host. Over time, a lot of the genes from the passenger here, the mitochondria, have been transferred to the host genome, so that now mitochondria cannot possibly live independently of the host cell, and the host cell is not entirely only itself. It is genetically partly mitochondria. So every one of the cells in your body is essentially what's called a chimera. It's a fusion of two different species. Actually, maybe many more than two different species. As you start studying the genome in any detail, you'll find all sorts of interesting invaders, including a lot of different viruses that have managed to insert themselves into our genome. The evidence supporting the idea that mitochondria, and by the way also chloroplasts, are endosymbionts, or start out as endosymbionts. Symbionts are organisms living within, actually, the tissue of another organism, or actually, in this case, within its cells. Mitochondria and chloroplasts, other than the fact that they live within other cells, look an awful lot like bacteria. They have their own ribosomes, they synthesize their own proteins. The ribosomes are actually poisoned by antibiotics that attack bacteria, whereas your cells, except for the mitochondria, are not poisoned by these antibiotics. Cyanobacteria are free-living bacteria that look very much like chloroplasts. Not only in their basic structure, they have internal thylakoid membranes, which are a critical structural part of chloroplasts, but they also have the same pigments that they use to gather energy. They contain chlorophylls A and B. That strongly suggests that the chloroplast used to be a free-living cyanobacterium. And finally, interestingly, mitochondria and chloroplasts both still contain their own genomes. They're circular in form, like the genomes of bacteria, and even though they don't contain all of the genes necessary for either of these organelles to live independently, The sequences of those genes are very similar to the sequences of proteobacterium genome and chloroplast genomes, respectively. The story gets even a little bit stranger than this, though, when you look at the phylogenetic relationships in the protists. So if you look at the nuclear genes, the genes within the nucleus of different protist groups, what you get is a phylogeny that looks something like this. It's not particularly well resolved. We don't know, for example, the relationships among these 6 different branches. But you can see here, and what I want you to notice particularly is that these names that have red or green colors associated with them are spread pretty broadly across this whole phylogeny. If you sequence just the chloroplasts in these organisms that are shown in the red and the green and the black, what you get is this. All of the red taxa, the taxa shown here in red, cluster together. These 2 taxa shown in green are also within this larger clade here. In other words, the relationships that you get when you sequence the nuclear genes are completely different than the relationships that are inferred when you sequence the chloroplasts. How is this possible? Well, it's pretty complicated. We've already talked about how the original chloroplast was probably a cyanobacterium that was engulfed by a larger cell and the 2 cells together became a photosynthetic eukaryote, probably something like a modern green alga. What seems to have happened since then is that some of those green algae became engulfed by other cells, so that the chloroplast within that eukaryote became a passenger within a still different cell. And of course, along with that chloroplast came its genome, and because it was being consumed by a cell that was totally unrelated to its original host, this chloroplast genome became lodged in an organism that was on a completely different part of the protist phylogeny. This actually seems to have happened several times. A photosynthetic passenger is a very useful thing to ingest and keep a hold of. In fact, there are animals that will ingest algae today and keep their chloroplasts alive within their own tissues to generate photosynthate. Presumably, it's something like this that led to these chloroplasts showing up in the brown algae and diatoms, the dinoflagellates, and the euglenids. Originally, presumably, the first endosymbiosis that generated a chloroplast happened over here in the clade that has led to the green and red algae as well as the land plants. But since then, the invention of chloroplasts has been adopted widely across other clades of protists.